like to begin by thanking the organizer for the opportunity to present some of our latest work. Today I'm just briefly introduce you how we use aptamer, a chemical antibody, to in vivo, to do targeted delivery, drug delivery, in vivo target cancer stem cell. We're using, the example I'm going to give you today is the one we target siRNA in vivo into cancer stem cell. Aptamer actually also has a nickname of chemical antibody. It actually functions very similar to the traditional protein-based antibody. In terms, it actually can bind a target very specifically and very tightly. Now, the way it works is we can actually use the example of tRNA. Now, it is actually consists of a single strand, must be single strand DNA and RNA, because we borrow an example of tRNA, right? If you have a single strand RNA or DNA, you will have internal base pairing, so you have a secondary structure. And it will then fold into a tertiary structure. Then because of this tertiary structure, it's just like an antibody, it has specific conformation. It will bind to its target. So the way of aptamer binds to its target, it actually in exactly the same way as how antibody bind to antigen. So it is basically, it's an induced fit interaction, shape fitting, right? Okay, so the way, now the aptamer are actually isolated using this so-called CLEX procedure, which they were published in 1990 by two independent groups in US. So basically, you synthesize, you start by a large single-strand DNA library, and you fold them into so-called aptamer. Then you're using, so you have a protein, you go there to fission, right? So you go there, you have your target, and you let them bind, and you wash off the unbound aptamer. So you have the bound aptamer, so, and then you PCR amplify this bind species. But after, say, 8 to 10 cycles, you can actually clone your aptamers, and you can then characterize them. Now, for targeted drug delivery in vivo, it does have some very important advantages compared to traditional monoclonal antibody. Now, it actually is it's very stable, right? The main thing for cancer, targeted drug delivery at cancer, the most important thing is it is because it is a very short stretch of DNA or RNA, about 20 or 15 nucleotides. So it is very, very small. Now, the size of a monoclonal antibody is about 150 kilodalton, right? Now, the aptamer we are working with is about 6 kilodalton. So it is about 25 times smaller than a monoclonal antibody. So in vivo, we know that when any agents get out of the blood vessels, it actually moves in our tissue by random diffusion, especially in cancer, in tumors, right? A random diffusion, the speed of any species travels, right? The speed is inversely related to its size. So because aptamer is a lot more smaller, so it actually penetrates, it moves much faster. It can actually penetrate tumor much, much better. So we just published a paper to demonstrate that, which I don't have time to show today. Now, the other thing is that because once you know the sequence, basically you can chemically synthesize them, just like we order in PCR primer. So it is very convenient. It is actually also being approved by FDA in 2004 to be used as a drug. Okay, so for today, I'm going to show you how we target cancer stem cell. Now, we know that in cancer, there are two groups of cells. One is non-cancer stem cell, the other is cancer stem cell. Now, cancer stem cell, they are very resistant to drugs. To my knowledge, on market, there's no single drug can actually eliminate, if very effectively eliminate cancer stem cells. And cancer stem cells, they are also very resistant to radiotherapy. That's why we can't cure cancer in most cases. So now, today the example we give you is where you target survivin. Now, survivin is known to be responsible in a lot of cases for chemoresistance. because survivin, it actually expresses very low level or not at all in normal cells, but it expresses very high level in tumor cells, especially when a patient has been treated with chemotherapy and radiotherapy, then survivin is overexpressed. Now, obviously, the function of survivin is anti-apoptosis because it keeps the tumor cells survive. So, we're going to deliver siRNA against the survivin. Now, the way we target cancer stem cells, we target, today the example I'll give you is target this EpCAM, epithelial cell adhesion molecule. Now, EpCAM is actually a cell surface marker for normal epithelial cells, but the expression level of EpCAM in normal epithelial cells is very low. Once a cancer derived from epithelial cells, the expression level goes high, very, very high. In breast cancer, the example I'm going to use today is we know that EpCAM expression level in breast cancer actually is 1,000 times higher than adjacent normal breast epithelial cells. Now, it has been shown a few years ago for breast cancer cancer stem cell, one of the set of cell surface marker is EpCAM positive, right? CD44 positive and CD24 negative. So we are going to targeted deliver drugs, and in today's case, siRNA into breast cancer stem cells using EpCAM as a target. So we actually developed this RNA aptamer against EpCAM. So it starts with a long molecule which truncated into 19 nucleotides. So it actually binds to EpCAM very effectively. So our design is how to deliver siRNA in vivo effectively to cancer stem cell. So the way our hypothesis is now we actually have our EpCAM aptamer. So this is as a guiding tool, right? It actually is covalently linked to the surviving siRNA. So our hypothesis is if we inject that into the mouse tail vein, it can travel to the tumor. It exists to the tumor, and it binds to the very highly expressed EpCAM on the tumor cell surface. Then it will undergoing endocytosis. Now, by yet unknown mechanism, once this chimera, so this is the reason we call chimera is it is actually two molecules linked together, right? So it's aptamer, targeted aptamer with the effector siRNA. Once it reaches the endosome, it will be released to the cytoplasm, and then it get out. It actually can to downregulate the target siRNA. So, so what we actually did is we have this our RNA aptamer target EpCAM. We actually chemically linked to the surviving siRNAs. This is published siRNA, surviving siRNA. It's not something we isolated. We just use this as a proof of principle tools to show that our strategy does work. So the model we use is a human, very commonly used model, MCF7, human breast cancer cell line. Now, if we incubate MCF7 with low dosage of doxorubicin, 300 nanomolar, for about 6 months, we can obtain a drug-resistant MCF7 variant. Now, it overexpresses EpCAM, surviving, right? So the wild type MCF7 expresses low level of surviving, just like in breast cancer. Now, once we treat these patients with doxorubicin, a chemotherapy drug like doxorubicin, the surviving expression will go up very about 15-fold. So this is a model, right, to mimic chemoresistance. So as you can see, it actually, if you look at IC50, this drug-resistant cell line actually have 5-fold increased resistance to doxorubicin. So our hypothesis is if we can knock down surviving, then we can treat, then we should increase the chemosensitivity of these drug-resistant cells to the normal treatment. So first, as I said, we use this chimera. So it is our EpCAM aptamer linked to a surviving siRNA, right? So this is an in vitro experiment. So in the cell culture dish, we have the MCF7 drug-resistant cells, right? All we do is we actually add this chimera, this RNA. There's no lipofectamine, there's no transfection reagent whatsoever. All we add is just this long stretch of nucleic acids, right? So our hypothesis is it should bind to the cell surface EpCAM, endocytosed, and go down, knock down, surviving. So this is what happens, right? So this actually shows, this is a Western, this shows actually it knocked down the surviving mRNA and also knocked down the surviving protein, right? So this is in vitro. So the next thing is we're going to generate an in vivo construct. Now, in order to do that, as I said, our construct is actually very small. This piece of nucleotide is very small, right? It's about 2.5 nanometer in size. Now, in kidney, we know the kidney filtration threshold is 10 nanometer, which means anything smaller has a size smaller than 10 nanometer. Once they pass through kidney, it will be filtered out, which is not very good for our treatment. So what we do is we actually add an PEG, so make it bigger. So now it becomes, after we add a PEG, about 15 nanometer in size. So our hypothesis, once again, is this, our aptamer carry this siRNA. You inject the tail vein of the mouse, which carries the MCF doxorubicin-resistant tumor. It should be able to travel to the tumor and get out and go there and knock down the surviving within the tumor. Now we are trying to see whether it is going to work in mouse tumor model or not. So first we're going to see whether it can indeed travel there. So this is an in vivo imaging. So the mouse has been anesthetized. This is a mouse which carried an orthotopic breast cancer model, MCF drug-resistant model. We inject one dose, about one nanomole, right? And we serial image it because our aptamer actually is fluorescently labeled. So basically, you can see it's actually after 4 hours, it reaches a very high level and gradually comes down. So it has very good, in terms of treatment, a reasonable good pharmacokinetics. Right? It lasts reasonably long in blood, but what we actually look at is the tissue pharmacokinetics. It's not the blood pharmacokinetics, right? Because the results I'm presenting here is the fluorescence concentration in the tumor versus time after one single injection into the tail vein. So the next thing is we want to demonstrate it to test whether our chimera, when inject to the tail vein, when reach tumor, can it actually knock down the surviving in this drug-resistant breast cancer? The answer is yes. So this is a Western, this is a quantitative, so it can knock down mRNA and knock down a protein. And actually, it is knocking down is actually indeed, it is due to the siRNA function, because you can actually sequence the surviving mRNA to demonstrate this action. It is actually by siRNA, because if one inject, you can inject siRNA, but it can also non-specifically activate microRNA pathway, because microRNA and siRNA using the same protein machinery, right? So if one activates microRNA, you can also non-specifically chop your targets, but at different places, right? But through this actually in vivo, sequencing the target surviving mRNA, we actually demonstrate it is indeed. Now this cleavage, this downregulation of surviving in vivo downregulate surviving mRNA is due to this siRNA. So this is the efficacy study. So we look at these mouse, there were about 6 mouse per group. So if we treat the mouse just with saline, so the tumor volume goes up very, very fast. If we treat the mouse with only doxorubicin, it just, the tumor grows delayed, but eventually it also reaches to the, to the very high volume we have to sacrifice, right? So this is actually a drug-resistant model, doxorubicin drug-resistant model, right? Now, it's the same mouse. If we knock down, in vivo knockdown using this construct to knock down surviving, then, so this is 3 treatments, right? We knock down, inject this chimera to knock down surviving first, and then 2 days later, we inject doxorubicin. So we repeated only 3 treatments altogether. Right? Now, once we knock down surviving, you see the tumor actually that grows much, much slower. It suggests that we actually make this drug-resistant tumor more sensitive to this, the same dose of doxorubicin in this control. The mouse actually survives much longer. So whether we have targeted cancer stem cell in this model or not, so the next few slides to demonstrate, provide some experimental evidence. So the first thing is after we in vivo treat the tumor with not this in vivo surviving, right, siRNA, and also with the same dosage of doxorubicin compared to the control. You see, if you look at the percentage of breast cancer stem cell population, right, in untreated about 12%. If you treat it with doxorubicin, it's also 12%. This match perfectly with previously, there are almost 100 papers from different groups show both preclinical study and clinical study that with very few exceptions, right? Medicine always have exceptions, right? Doxorubicin does not kill cancer stem cell, right? So this is exactly what we observed. But now, if we knock down surviving in vivo, then we use the same dosage of doxorubicin, which is 5 milligrams per kg, we actually significantly reduce the percentage of human breast cancer stem cell percentage in this model, right? So now we also show, this is just very busy slides, but basically after we treatment, we actually take the tumor, we make single cell suspension, and we sort this treated tumor into a single cell suspension. But basically this confocal study show we provide the physical evidence, right? By inject our construct into the tumor in the tail vein, our construct get into cancer stem cell in vivo. So basically, without going too detailed, this is the quantification. So basically, with all those slides, it also increased the chemoresistance. It kills a lot more cancer stem cells than the free doxorubicin and control. So basically, let me just, so we, for the first time, this has just been accepted, this paper. So we show that we can actually very effectively in vivo targeted deliver siRNA to cancer stem cell. Thank you.